Now we're going to get into chapter 3, epigenetics. We touched on epigenetics a little bit at the end of the last chapter, but what I want you to understand ultimately that epigenetics is what makes our lifespan pretty unique. Our genes, of course, are going to help with this, but epigenetics kind of asks the question, what other factors may play a role? We don't have to dive too deep on this, but DNA methylation is important. It results from a methyl group that is attached to a cytosine. And ultimately, methylation renders the gene inactivated. I want you to remember this. Methylation equals inactivation. In science talk, the genes are then transcriptionally inactivated. This process is going to play a very prominent, important role on controlling how cells are activated, and therefore it plays an important role in human development and the diseases that affect us. Histones are another important piece of epigenetics. Histones are proteins. They help pack that DNA into the nucleus of the cell. Chromatin is also involved. It's the strands that give the nucleus its granular appearance. But in histone modification, there are chemical changes of the histone proteins around which the DNA is ultimately coiled to pack that DNA into the nucleus. So there's interaction between these two, the DNA and the histones. So if there's some mutations going on during the modifications of the histones, there can be a change in the nearby gene expression. This modification is critical for normal development, and mutations can really impair this process. So congenital heart disease is an example of histone modification that has been impaired by mutations. That's important to know. So in the first question here, the epigenetic process that results from attachment of a methyl group to cytosine is which one? The answer is B. That's the definition of DNA methylation, the attachment of a methyl group to a cytosine. So how does epigenetics work in human development? So you have to think we as organisms have stem cells. It's important to know that stem cells are what's called totipotent. They can give rise to all cell types. They have the ability to specialize in whatever they want to do within the organism. That's why stem cell research and manipulation is very controversial because they can make whatever type of specialized cell they want out of stem cells. Epigenetic modifications that happen during our embryonic development really help to make sure that specific genes are expressed in the correct types of cells and tissues, and other genes that should not be expressed are silenced or methylated. We also have what are called housekeeping genes. And you need to know about housekeeping genes that these cannot be silenced or methylated. They remain transcriptionally active in nearly all of our cells. They are not affected by methylation and they cannot be silenced. So knowing these things, in what stage of human development do you think that epigenetics has the most impact? That would be in utero when our stem cells are deciding what cells they are going to specialize into. So I touched on this and introduced it very briefly in the last chapter, genomic imprinting. We discussed in the last chapter that for every gene that is not a sex chromosome, a child will inherit two copies, one from mom and one from dad. Most of the time, both mom and dad's genes contribute to the phenotype of the offspring, or how the offspring looks. This is called biallelic expression, bi meaning two. Sometimes, though, gene expression is randomly monoallelic. At times, the maternal cell can be inactivated or methylated, and at other times, the paternal cells are inactivated or methylated. Even more rarely, if there's imprinting of a maternal or a paternal copy, that means that the gene is inactivated or silenced from mom or from dad, and it remains inactivated in their children on the sperm of the egg or egg of the offspring. This imprinting can affect the size of the offspring. If it's maternal imprinting, meaning that the gene is coming from mom, the silence gene can be coming from mom, it can reduce the size of the offspring. And if it's coming from dad, sometimes it increases the size of the offspring. This genomic imprinting can also contribute to certain syndromes. Again, there are differences here depending on who the silencing is inherited from. Prader-Willi and Angelman syndrome are two examples of genomic imprinting. They are both due to a deletion on the long arm of chromosome 15. But you need to know the differences here. If the imprinting or inactivation of the gene is inherited from dad, it's known as Prader-Willi, and you can see the characteristics on your slide. If it is inherited from mom, it is known as Angelman syndrome with totally different characteristics. So here you can see a child with Prader-Willi syndrome on the left and Angelman syndrome on the right. This is a visual of the genomic imprinting. This is in your book. What I want you to notice here is obviously it's all on chromosome 15, but it depends on whether it's coming from mom or dad. So on the left, you can see Prader-Willi syndrome. You can see the deletion is on chromosome 15, but it's on dad's chromosome. And on the right is Angelman syndrome. The chromosome deletion is also on chromosome 15, but it's coming from mom to result in Angelman syndrome. Another known example of imprinting is Beckwith-Wiedemann syndrome. This syndrome is associated with phenotypes where there is typically body overgrowth. What I want you to know about this syndrome too is that it's accompanied by an increased chance for cancer. 20 to 30% of those who have this syndrome is caused by an inheritance of 2 copies of the chromosome from the father and no copies from the mom. Her genetic information is completely missing. This syndrome is usually identifiable at birth. Babies are usually large for their gestational age. They may have hypoglycemia as a neonate. They may present with a large tongue. They'll have some creases on their earlobes and possible defects of the intestines. As far as the cancer that they are increased risk for, a Wilms tumor on the kidney or hepatoblastoma, which is a tumor on the liver, are risk factors for them. Imprinted genes are not the only reason for epigenetic modifications that persist over time. There are conditions that are encountered in nature during our childhood and adolescence, and even later that can have long-term impacts on our epigenetics as a whole. Some of these impacts can be transmitted across generations. Nutrition is an example of that. Our book tells us a story about the winter of 1943 when there were millions of people in the Netherlands who suffered from starvations as a result of a Nazi blockade. So that interfered with the shipping of food. This was called the Dutch hunger winter. Researchers found out that individuals who were in utero during the Dutch hunger winter were more likely to suffer from obesity and diabetes as an adult, compared to their counterparts who had not experienced nutritional deprivation during gestation. It's largely unknown, though, how this works molecularly. But what we do know, and that what you should know too, is that epigenetic modifications in utero related to nutrition deprivation can lead to higher incidences of diabetes, obesity, and cardiovascular disease. It makes sense then that maternal care as a whole can affect epigenetics. Fetal alcohol syndrome is an example of this. When there is alcohol or ethanol exposure in utero, it can impact DNA with our skeletal and our neural development. It may repress fetal expression of certain DNA. In other words, alcohol impairs neural stem cells becoming functional neurons. So let's do a question here. Genomic imprinting is best described as which one of these? The answer is C. Some genes are silenced depending on which parent transmits them. For some genes, some are transcriptionally active only on one copy of a chromosome, maybe the copy inherited to the father, or maybe the one inherited from the mother. then the opposite would be transcriptionally inactive. This is gene silencing or imprinting. To further drive the point on epigenetics and how outside forces affect DNA methylation, scientists study identical twins. That provides them opportunities to study the impacts of epigenetic modifications, especially with aging, different lifestyles, and the effect of the methylation patterns that result. Finally, I want to touch on epigenetics and cancer. So, the study of epigenetics and the modification in human disease largely comes from studying cancer. What I want you to know here are there are two abnormalities in the DNA of tumor cells that they have found. One is that there is hypo or low methylation in the genes that promote cell division. So, what does that mean? If there's low methylation in the cells that promote cell division, it means that cell division is not typically silenced, maybe where it should be, and the cancer is able to divide rapidly and abnormally. So normally these genes would be methylated, but because there's low methylation or hypomethylation, they are not, and the cell is able to divide and divide and divide, causing a problem. The other abnormality here is that there is hypermethylation or too much methylation or a lot of methylation in tumor suppressor genes. So what does that mean? It means that the tumor suppressor genes are being highly methylated, meaning that they're getting turned off. So what does a tumor suppressor gene do? Just how it sounds. It helps keep cell division in check. In other words, those genes are silenced because they are methylated. I want to stress here more and more, we're realizing that the environment plays a huge role in epigenetics and therefore the development of cancer as well. We see this in the BRCA1 gene, for instance, something that doctors can check as an inherited breast cancer gene. The good news is they're making advances in epigenetic screening and are constantly coming up with less invasive, less expensive, and less risky ways to screen for certain cancers like for the breast, colon, bladder, and prostate. There are also some advancements in treatment of epigenetic diseases. They are finding that sometimes epigenetic modifications can be reversible. DNA can be demethylated, and the histones that we talked about in the beginning of the chapter can also be modified. An example of demethylating agent is this azacitidine 5. It's a type of chemo, and it utilizes complementary nucleotide bases as a demethylating agent. So if it is demethylating, it means it is turning things on. So know that that's been shown in a promise in treating pancreatic cancer, which can be very devastating and lead to an untimely death. So important to know that they use that for advances in pancreatic cancer.